Hi, it's Miss Barnhart, and today let's continue our study of genetics by learning how to solve genetics problems. We use the Punnett square, which was diagrammed here. Before we begin, let's review the essential vocabulary terms from the first part of the lesson. Please make sure that you can define trait, gene, allele, dominant, recessive, genotype, homozygous, heterozygous, and phenotype. If not, please pause the video and record these definitions. We will use capital letters to represent the dominant allele and lowercase letters to represent the recessive allele. Big A represents the homozygous dominant. Big A represents the heterozygous. And little a represents the homozygous recessive. Note that big A and big A will have the dominant phenotype, while little a exhibits the recessive phenotype. The Punnett square is a diagram that is used to predict the offspring of a particular cross or breeding experiment. The diagram is used by biologists to determine the probability of an offspring to have a particular genotype. Here are the steps I encourage you to use as you work through the problems. Set up a key, clearly identifying the dominant and recessive alleles. Identify the parental genotypes from the information given and write these down. Determine the gametes that can be produced and write these across the top and side of the Punnett square. Fill in the possible zygotes. Determine the genotypic and phenotypic outcomes. Remember that the results of the Punnett square are predictions. We can test the validity of these predictions with the chi-square test. You'll find that with practice, different parental crosses can result in different phenotypic outcomes. Check these out. Here's a cross between two heterozygous parents. Note that each parent will give one of his or her big A or little a alleles to the gamete. These possible zygotes are written to the top and sides of the Punnett square. The zygote combinations are then recorded in the boxes of the Punnett square. You can see then that these three will express the dominant phenotype. You can see then that these three will exhibit the dominant phenotype. The genotype ratio is 1 to 2 to 1, but the phenotype ratio is 3 to 1. This is what Mendel found in his experiments with the pea plants. In the second cross, a heterozygous parent is crossed with a homozygous recessive. Like in the previous cross, record the possible gametes for each parent along the sides and top of the Punnett square. When the zygote combinations are recorded, filling in here, you can see that there is a 1-to-1 ratio for both the genotype and the phenotype. But what if we don't know the genotypes of the parents? After all, the homozygous and heterozygous dominant individuals have the same phenotype. One thing we can do is a test cross. So here, to determine whether the purple flower plant is homozygous or heterozygous for the purple allele, we cross it with a homozygous recessive plant, which is a white flower. If any of the offspring are white, then the plant has to be heterozygous. If all of the offspring are purple, it is likely that the plant is homozygous. A dihybrid cross involves inheritance of two different traits. If the genes governing the inheritance of these traits are on two separate chromosomes, then there's going to be 4 possible combinations of gametes in the offspring. Remember independent assortment here. Note that big A, big B, little b can give rise to 4 combinations. We get big A, big B from the first of each allele pair, the big A and little p from the outside of the genotype, the little a and the big B from the inside of the genotype, and the little a, little b from the last of each allele pair. This is the FOIL method. Remember that in a dihybrid cross, your gamete must have 2 alleles or letters, one from each gene trait. Now let's take a look at an example. In peas, round seeds are dominant to wrinkled and yellow seeds are dominant to green. Let's examine the cross between 2 plants that are heterozygous for both traits. The gametes that can be formed are big R, big Y, big R, little y, big Y, and little r, little y. You'll see that the gametes were filled across the top and sides of the Punnett square. They're identical in this case since the parents are both heterozygous for both traits, and the genotypes for the offspring are completed and color-coded for the phenotype. Can you see they're 9/16 round yellow, 3/16 round green, 3/16 wrinkled yellow, and 1/16 wrinkled green? My suggestion is to practice many of these basic crosses so you get used to the process. Now, until next time, stay curious. I hope that's helpful.